Good morning, everyone. I have to make an official start because as soon as I hit these buttons, the timer goes on my slides. And it's the same for every speaker, and I'll explain in a moment. So here we go. Okay, good morning again. My name is Mark Graham, and I'm the Vice President of Research and Collections. Welcome here today. We wanted to have everyone together from the museum to tell you a few things about our department and to keep you updated. We've had some meetings last week to discuss our planning for next year, so to get an idea of the budget things, but also how we want to work together with all of you. We realize that the things we do are so interconnected with the parts of the other parts of the museum. We're using this format, Ignite, and it's a prescribed format, just like you're seeing right now. It's meant to be simple, short, and deadly interesting. And I'm sure it will be today. As you can see, you will see there are 20 slides for every single speaker, 15 seconds per slide. So after 5 minutes, you have a deadly interesting presentation. So that's it happens every 15 seconds like that. We're going to be talking about research and collections, which is made up of collections in plants, animals, minerals, and fossils. And so that's it's also, you'll see in a second, that's also the way we organize ourselves, but it's not all. If you have that kind of thing, you also are interested in intellectual property, photos and documents, registering all those things, conserving them for a long time so people have access to them, but also all the information in our library and archives that describes them. This is the way we organize ourselves. So we have a section on a group of people working in paleontology, mineralogy, botany, zoology, and CSIM, Collections, Services, and Information Management. So I'm going to spend a little time telling you about our collection-based science program. All of our workers are either helping to loan out, take care of, do research on, make some sense out of those invaluable collections that we have. We have two areas that we're very proud of and very good at. We have a lot of work that goes on in the Arctic. And so that region of Canada gets a lot of attention from us, but also species discovery is something that happens in all of those areas. Understanding what are the new species, describing them, etc. The way we go about that is through programs of systematics research. And there are lots of components involved in systematics research, and I'll spend a little bit of time telling you about each of those. So part of that, a big part of that is taxonomy. Like Richard Owen here, the founder of the British Museum, you name new species, like that flightless bird, and you find systems of classifying them. How do they fit into the scientific system of classification? If you get that kind of work done, oops, see, we're out of sync. You also think about biogeography, where those things live, either in general terms or as a species. We also think about how they're related to each other. This is really interesting. Systems of phylogeny, how closely things are related, like those lobster species, or a whole bunch of plants and animals on a whole different scale, and talk about how long things took to evolve to get like that. How do we do that? We work in the field, so we go out and actually collect things. We work in labs. It's a lot of the time spent actually analyzing those things later. Lots of analyses. Everything we do is in collaboration. Some of the end products, a lot of the end products, 10 million plus, are part of an authoritative collection, things that we know have the right names, geographical references attached to them, etc., like those crustacea. Some other products that we have, primary publications in science journals, communicating with our peers, also in books like that wonderful book that Donna just published, editorials in newspapers or science journals, blogs, and reports. We also give lots of advice with our expertise in governing boards, editorial boards for those magazines and publishing things, professional organizations, which each of our disciplines usually has and needs expertise in running, but also government organizations. We apply ourselves in lots of educational ways, in exhibits, lectures, with interns that need mentoring. Also, Students on Ice gets our expertise, but we also supervise lots of graduate students and spread that information to the primary and secondary student level. That's all I have to say. 5 minutes goes fast. We have 11 more talks. I have a few more seconds. And our first speaker is going to be Kieran Shepherd. Wow.
Hello, my name is Kieran Shepherd. I'm the curator of the Paleobiology collection at the Canadian Museum of Nature, and I hate dinosaurs. Yes, it's true, I hate dinosaurs. As a young boy, I collected lots of stuff: fossils, minerals, bugs, bones, just about anything that I thought was important. My love of the museums, the natural history stuff, was first developed at the Royal Ontario Museum, where as a young boy I made weekly trips to the museum with my dad. I decided I wanted to work in the museum, so I collected a whole bunch of paper at a variety of universities. When the time came, I ended up at the museum. Dinosaurs are a major component of our paleobiology collection and with world significance. I just came back from the annual meeting of the Society of Vertebrate Paleontology. Of the 19 or so dinosaur talks I attended, 11 featured CMN specimens. But a collection is so much more than just dinosaurs. It is a world-class collection reflecting the diversity of life in the vertebrate past. Almost entirely Canadian or Canadian-like. Geology knows no boundaries, and about 50% of it's from the north. One of my favorite parts of the collection is that of the tertiary mammals, that magical period of evolution after which dinosaurs finally got what they deserved. Animals such as Corythodon is one such cool, sexy beast, just as interesting as any dinosaur. And there he is. What is sexier than this thing? Even Canada Post, dedicated a stamp to this lovely creature. One of his features is he has the smallest body-to-brain ratio. In fact, it's one Baby Bell cheese per head down for 500 grams. There we go. Anyway, oh dear. Dinosaurs were always sort of a pop culture. They were associated with natural history museums, grade B movies, and of course, toys. But the level of public interest and passion and weirdness directed toward dinosaurs increased dramatically with two events. One being, of course, this stupid movie, which occurred in 1993, and I reluctantly did not go to this movie for several weeks. After that, it was just downhill for our society for this cult-like behavior about dinosaurs. TV shows, films, Discovery Channel, BBC developed all these specials that continue today. And anyway, what need I go on? Another weird event, of course, that occurred is Barney and Friends. Many of you grew up, I'm sure, as kids watching this stuff. This spawned a scary amount of dinosaur-related stuff, and there was no escaping dinosaur fever in the 1990s. Not even our trusty pets and beverages were immune from dinosaurs. Even one of my favorite TV shows was invaded by dinosaurs, and it was at that point that I just gave up. And I had to admit that these freaks of nature, of the natural world, I had to embrace. So I went to my first encounter with dinosaur culture was at Meet the Experts, or as I like to call it, Cornered Curator. Here at Meet the Experts, it changed my opinion about dinosaurs and their role in society and the museum. They inspired our visitors to ask really interesting questions, and they made equally interesting observations. Nothing tests the scientists more than being on display and exposed to the public. And some of my top 10 questions, and these are real. Could dinosaurs vomit? Mr. Shepherd, I don't have a question, but I have a comment. Were some dinosaurs omnivores? Who killed the dinosaurs? Did male dinosaurs have... Next question. Read that book. Could dinosaurs swim? Could dinosaurs talk? Did dinosaurs smell bad? Why are dinosaurs not mentioned in the Bible? That was a trap. Why are people who study dinosaurs so weird? It's obvious. I also got involved in my first dinosaur project, which was truly a rewarding endeavor with Robert Holmes and Dr. Michael Ryan, who are research associates. A truly rewarding endeavor, a legacy of which, of course, still lives on today in our fossil gallery as far as Chasmosaur urbanensis. I also, a few years later, was involved in the construction of our new fossil gallery, which, as you know, includes lots of wonderful dinosaurs. Made me reflect on past galleries and the effort to build such monuments to past life. Whether we like it or not, dinosaurs fill our museum. They make a natural history museum accessible to both young and old, and of course, they're a moneymaker. Dinosaur research is a hook, but for me, the biggest selling point of dinosaurs is they inspire our youth into lifelong interest in science. While most outgrow their passion for dinosaurs, their interest in science leads to careers in biology, geologists, or just members of the public who really support natural history museums. Thank you.
Good morning. For those of you who don't know me, my name is Alan McDonald, and I've been with the museum for about 15 years now. I work in the paleobiology section with Kieran, and I've been asked to talk to you about what I do today. First off, I'm your fossil preparator. Simple enough. I work with fossils. No, not like these guys. Real fossils. Here we have a fish, marine reptile, plesiosaur, and another fish. I take the various chunks of rock and with fossils in them and try to bring them to the light of day. Wrong slide. I work on fossil material containing primitive fish, aquatic birds, marine reptiles, dinosaurs, and of course small mammals. These animals may have died from old age, from being attacked and or eaten. What's the next one? From natural disasters or from some freakish accident. And somehow got fossilized. Later, millions of years or even just a few millennia, paleontologists like Steve Kumba go out to Saskatchewan and Manitoba into the woods and the winding waterways to find these lost treasures. We are looking for, preferably, fossils in situ in the strata of riverbanks, but most of the time, because of washouts in the spring, we find them in the tumble down along the riverbeds. Back at the lab, we'll try and prepare them one of two ways. I'll usually either do them mechanically prepped using hand tools or with acid prep using acetic acid immersion. This is the acid prep lab. With the bone beds, typically we'll do them in the acid prep lab because the nature of the bones inside them, there's so many and they're so delicate that it's more conducive to do them without using hand tools. The bone bed blocks are first immersed in the water and then allowed to soak in acetic acid emulsion, which is a combination of acetic acid and water. And then months later, or even years, you'll end up with all the matrix dissolving and a multitude of either vertebra, teeth, or even coprolites, which are a big deposit. I tried. Anyways, larger specimens I tend to mechanically prepare. Most of my tools are the stuff that your dentists use, dental picks and drills and stuff like that. Larger specimens like dinosaurs can take years to prepare, up to several. Now I'm lost. Here we have an as-found fish block from the time we open it up in the lab to the bottom right-hand side is the final specimen as I finished off. Sometimes the blocks are fractured when we get them, and they will show cross sections of the bone along the fracture lines. So sometimes we'll prepare them separately just so we can analyze the cross sections later. And then you can obviously see it matched up with its counterpart there. This is a fish, a large chunk, 4-inch chunk of fish skull that I've been preparing over the years. There's a number of pieces to the skull, and it's just a huge skull. After all prep is done, I tend to help the paleontologists with illustrations. I'll detail map locations, I'll organize photographs for them, and I'll also illustrate specimens. I'll illustrate using either the Adobe Photoshop program and the Illustrator program, or by hand, so that I can offer a wide variety of options for the paleontologist. And just recently I went to the Carnegie Museum in Pittsburgh, where I learned from their staff some very good casting and molding techniques that we're hoping that I can start up a small program here doing the same. And that's about it. Most of the time, people ask me why I got into this. Lots of friends and family have helped me along the way, and for the most part, it was my grandmother. I always told her I wanted to be a paleontologist when I grow up, and for the most part, I guess working with them is okay. Thank you.
When I told Laurie Baudouin the title of my talk, she said, "Well, that's interesting." So I've got 5 minutes to change Laurie's mind. Here we go. We've been sitting here for 15 minutes. You need an exercise break, so this is the participation part of my talk. I'm going to ask some questions. When you answer them positively, raise your hand, keep it in the air. Here we go again. If aquamarine, topaz, tourmaline, moonstone, emerald, or peridot, or alexandrite are your birthstones and you have one, raise your hand in the air, keep it there. Come on. If you've cooked with glass pots, if you've had a dental crown in the last 5 years, if you use a cell phone, if you've had a medical or a dental x-ray, and if you've been caught playing with a PS3, raise your hand. Now take a look around you. Congratulations, everybody. You've just proved in 2 slides how important pegmatites are. Those things come from granitic pegmatites either exclusively or mostly. So you use granitic pegmatites in your lives all the time and you didn't even know it. So what's a pegmatite? Take a look at the picture over here. Pegma comes from the Greek word meaning interlocking. It's that texture that you see on the rock. If you see that texture, you're looking at a pegmatite. But as in science, things get popularized and get taken away from us. And today people think that pegmatite means any rock with big crystals, just because pegmatites have giant crystals. So you're even going to hear people like Paula Phelan claim that she works with pegmatites too. What about museums? Why should somebody like me in a museum work with pegmatites? Well, one of the reasons is we're one of the biggest clients of pegmatites. A lot of those pretty minerals that you see on display come only from granitic pegmatites. So wouldn't you like to have one of us look at these things? So that's it for part 1 of the talk. Part 2 is, I've told you about pegmatites, now I'm going to inject myself into the talk. What do I bring to the table? What do I study? Here's a list of just some of my activities. And yes, counter-espionage is up there. I spend a lot of my time, a disproportionate amount of my time, working with minerals of tantalum. It's a very, very expensive mineral. It's very high in its commercial value. It's clean to the environment. And in tropical climates, it's very easy to extract. And it's largely because of tantalum that the Rwandan army this summer was able to invade the Congo. Rwanda's got large deposits of tantalum that actually lie loose. They don't even have to bring in heavy mining equipment. And this invasion has been largely funded by the ability of Rwandans to get tantalum. I spend a lot of time working on the classification of minerals. This slide that I've got up right now is a first attempt by me, came out in 2005, to show geologists how to put a name on a species even when its structure is thoroughly destroyed by radiation. I spend time classifying environments. Rocks, magmatic rocks, are not really a species, they're an environment within which mineral species occur. This paper I did with Peter Cherney is one of the top 20 papers of all time cited in Canadian Mineralogist. I spend time characterizing crystal structures. These 2 minerals, muriatite and sesquitertantite, were minerals that I first saw the structure of, and today these are being investigated by physicists because of their properties, being able to trap large ions, and they're being used in radioactive waste storage containers. I spend time working on systematics, trying to understand how and why minerals behave the way they do, and even geological systematics, which is trying to understand what it is about natural environments that concentrate elements into groups. In the mid-1990s, I even got money from the Northwest Territories and Yukon governments to go up and find deposits for people of the North to work, and you know what? Lucky as we were, we found a gem deposit, and it's still being worked today. But right now I spend a lot more time working on something known as the Grenville Mountains. These were a massive Himalaya scale mountain chain that spanned the world's first supercontinent, Rodinia. I run around from different continents, and they're all broken up today, and I hop from continent to continent trying to study bits of it. In particular, I'm interested in what happened at the root of the mountain chain. We're talking about 30 to 40 kilometers deep into the earth. Rocks melt that far down in the earth. I'm trying to figure out whether we can tell whether rocks form metamorphically or igneously, and that may sound interesting, it may sound simple, but it's a very tough thing to answer. Counter-espionage, take a quick look at that slide. You're only going to see it for 10 more seconds. As soon as it disappears, you'll know that you've had the first look in Canada, in the USSR, in the world, at the actual location of the former USSR's first and biggest tantalum deposit. Not even Russians are allowed to post that. My last project, no, that's legit, ask me later. My last project is one I'm doing with the Smithsonian. It's a Google Earth application, and it's got applications from simple tourism right up to stuff that we geologists like to work with. And I went over. Ha ha ha.
Okay, so my name's Paula Pielanen. I'm a mineralogist with the research division, and I hope you're probably very glad that I'm not throwing things into the audience with a title like this. So today I want to talk to you about some of the work I do, and the alternate title for this talk could be Ugly Minerals and Why We Should Love Them. So Scott talked to you about pegmatites, and I promised him I would not use the P word, but we actually work on things, we work in the same kind of area. What he does on granitic stuff, I work on alkaline rocks. And in his rocks, you'll see a lot of quartz. I can't identify quartz, and that's the one main difference between what we work on is that I have no quartz in my rocks whatsoever. It's one of the first minerals that we all ID as a kid, right? We teach people to ID. In my rocks, if I see quartz, it's probably a mistake. So I work on things that are called alkaline rocks, and all you really have to know is that they're enriched in sodium, they're enriched in potassium, and they have very little silicon in them. So we use something called an alkalinity index to actually classify our rocks. You also have to know they occur everywhere. Although they account for less than 0.1% of the Earth's crust, they occur on every continent, every type of geological environment, but they dominate in places that have deep crust that we can have fractures right down to the mantle. So the red rock part of it, I'm going to talk to you about a project that we have going on in Thailand and Cambodia right now, and as you can see, the dirt beneath our feet is all bright red. It's laterite, and what it is actually weathered basalt that's been turned into soil. What we're looking at is the zircons in this basalt. So where they occur, and I don't have my laser pointer to show you, but you can see the yellow areas, that's where we're thinking that these alkali basalts occur, whereas opposed to normal basalts that we see occur where the red dots are. We're looking for zircons. They also mine sapphires and sapphires and rubies, which are corundum in these environments, and they form really, really, really deep in the mantle. So they're not a normal rock. They come from a deep part in the mantle that has a very different chemistry. So the basalts that we're looking at occur all over Southeastern Asia, in Cambodia and Thailand, lots in Vietnam and in China, and they're basically a result of the after effects of the Himalayas being formed. This is a mining that occurs, and this is in Cambodia, in Phum Tram. We were up there in January, and it's not the most highly sophisticated mining that exists. To basically dig a hole 30 feet and pull up the zircons from the basement and do it by hand. Basically, they're just shoveling dirt. My colleagues were smart enough not to let me climb down the hole, so I'm still here. What they're looking for are these little ugly, little red and brown zircon crystals that they actually take out of the alluvial material, out of the laterite, pan it, or just clean it with a hose, and they get these bright red to brown zircons out of it. And once they're done with that, that's a red mineral. They actually wind up turning these things with a reduction process to these bright blue gem materials. So it's actually uranium 6+ to uranium 4+ conversion in the atomic structure of the zircon that turns it to a bright blue color at about $44 a carat wholesale. So red mineral, blue mineral, why are we looking at them? Well, they're pretty for one. They make great collection items, make great jewelry. But what we want to look at is actually the chemistry of this mineral and try and understand how we can determine the process and the magmatic conditions under which it formed. And I do this for all different minerals, and not the pretty minerals that we mostly see on display, but usually the ugly minerals that everybody else ignores in the rocks. Zircons is zirconium silicate. It's used extensively in age dating, which translating, this is a word I learned last night. We can use it to figure out what the conditions of formation were. If there's zircon, we know that it exists at a certain condition, usually a high silicon activity. And if there's no zircon, then we know that it's usually an alkaline environment. So in my rocks, very rarely do I get zircon. They're refractory. They don't actually break down very easily. They have very good growth patterns, so we can just like looking at a tree ring, we can analyze the different growth rings and see what conditions they formed at. And for my purposes especially, we look at isotopes, whether they're oxygen, which they retain, or for age dating purposes. These are actually some of the Larvik plutonic complex zircons that we've looked at, and we can use them to tell us how old they actually are. I can take a breath. Okay. These aren't Cambodian or Thai zircons, but these are ones from the Larvik plutonic complex, and I'll use the P word here, but they're from pegmatites. But you can see that the trace element signatures are very different. Secondary ones and primary ones are 2 generations, so it's basically a fingerprint in trying to understand how the mineral formed, what the environment was like at the time that it formed. So the next steps in this project and for the Larvik project are trace element analysis, helium, hafnium, and oxygen isotopes, and apparently nothing else. So.
Okay, so my name's Jeff Sorella. I'm a botanist here at the museum, and I'd like to talk about the work we're doing on the Arctic flora, Arctic plants in the Canadian Arctic. So Lynn Gillespie and I are leading a project, co-leading research to study plants in the Arctic. Usually we hear about ice in the Arctic and snow, but the Arctic's really green if you go for about 6 weeks in June and July, and a bit of August. So if you're in a spaceship looking down at the Arctic, you'll see, well, a lot of ice up top there, but around the continental part of the planet is the Arctic ecosystem. Canada has about 36% of the global Arctic, so we have more Arctic territory than any other country in the world. So we have a real responsibility to understand the Arctic and to take care of the Arctic so that the Arctic can exist into the future. In the next 100 years, the climate is expected to change dramatically in the Arctic. This model is about 10 years old, so it's out of date. By 2090, there could be about 10 degrees Celsius warmer on average in the Arctic. This stuff is happening a lot faster than has been thought in the past. People have been studying plants in the Arctic for about 200 years. Some of the first plants were collected by the Perry expedition, 1819, 1820. 100 years ago, the Canadian Arctic expedition went up, collected plants in areas that we revisited earlier a few years ago. So the botany team at the museum has been conducting field work in these areas in the Western Arctic in the past 5 years, and we were in the Eastern Arctic this previous year that Paul's going to talk about. So we're trying to understand what are the plants that occur in areas, how do we tell them apart. All this is based on specimens, so we collect material in the field and bring it back to the museum. It goes into the herbarium where these specimens document the distribution of species in time and space. And the specimens are fundamental to understanding what these things are and that can be revisited by scientists in the future. Museum botanists like Erling Porsold have been producing seminal, the most important publications on Arctic plants through the last century. The problem with the flora is as soon as it's done, it becomes out of date, and science is progressing very rapidly. So we need to have a system to understand, and we need a resource that shows what are all the plants in the Arctic right now. So that's exactly what we're doing, a new project to produce a new catalog of the flora, all the plants in the entire Canadian Arctic ecozone and northern Alaska. This has never been done for the entire Arctic region because it's a big area, but it's important that we do this. So we've amassed pretty much all the international experts on the Arctic plants. There aren't very many. A lot of them are in Canada, but not all of them. We've had some meetings, and the project's about 1.5 years underway, and we're just starting to kick things off.
All right, that's my cue. My name is Paul. I'm a research assistant, and this sorry state is brought about by 24 days out on Nunavut collecting plants. The pictures speak for themselves, really. So this past June, Jeff, Lynn, myself, and Roger embarked on a collecting trip to Katanalik Territorial Park. Katanalik in Inuktitut means place of waterfalls, and truly it is a remarkable place with many beautiful sceneries like this. Lynn was the only one who didn't have a silly picture. Now, before we embarked on our trip here to the Soper River Valley, you see it as this green ribbon here in the southern Baffin Island, there were only about 60 collections in the museum's herbarium from this region. Many of them were collected by Soper himself, who the river was named for. Because it was a river, we actually got to paddle it. And so in our 24 days on the tundra, we actually covered 56 kilometers, 8 Class 2 rapids, in our sequel to 2009's Paddle to the Plants 2, or as I like to call it, Paddle Harder. When we weren't paddling, we spent most of our time collecting and pressing plants. Now, this actually hasn't, this is the same technique that's been used to preserve plants for hundreds of years, hasn't changed. And yes, we spent many, many hours in the pressing tent, carefully arranging the specimens from the herbarium. After our trip, we brought home approximately 900 collections, representing some 250 different species from this region. It's a low Arctic region, so it's incredibly diverse. Some of our notable collections for this region include, I forgot to say, we also collect in large amounts. We collect plenty of duplicates, and so our 898 collections actually become 3,500 herbarium sheets that are stored across Canada and around the world. Thus, every expedition brings back so many species. Some of them include bog rosemary, Andromeda polifolia. This is only the second time it's been found in the Canadian Arctic Islands, the first being found in 2008? Yes. The coral root orchid, this is the third collection that's been made for the Canadian Arctic Islands. This is a tiny, cute little plant. Strict primrose, this is the first time that we've collected it in the eastern Canadian Arctic. Previous to that, it wasn't known from there. And the most exciting collection, I think, is the northern bog orchid. This orchid, we found it by the hundreds. It was all over the place in this park. And this is actually the first time that it's been found in the Canadian Arctic Islands. So it's extremely noteworthy, and if we hadn't gone, we wouldn't know it's there. Thank you very much.
Today I'd like to discuss with you why Canada has, in the form of our National Herbarium, an enormous growing collection of plant specimens. Time is a bit tight, so your part of the discussion has been provided for you. Your lines were provided by your fellow citizens who have visited us in recent years. And like you, they want to share our pride in this great resource we all co-own. So they want to learn to love flat plants as much as we do. So to start with, plant specimens begin with an adventure in the field, in circumstances that are sometimes wildly thrilling, but that are at the very least pretty interesting. And those adventures are distilled on specimen labels that look like this. And the beautiful, weird, unexpected, life-sustaining plants that are collected on those adventures, at least those that aren't flat to start with, are made flat and dried to a crisp, resulting in, well, flat plants, but flat plants documenting not only adventure, but also many incremental discoveries, beginning with the discovery of that plant in that place on that fateful date. A profusion of information in just 2 efficient dimensions, so that in a relatively small space, we have over 1 million flat plants, lichens, and algae, and being dried, they last for literally hundreds of years. Flat is also a pretty convenient format for mailing, which is important because there are people all over the world serving the common good through projects involving our specimens. So it's actually like having over 1 million flat Stanleys that travel periodically on adventures of their own. When researchers use specimens, they sometimes attach labels that show where flat Stanley has been and what he was useful for. The more research projects, the more a specimen's value is realized. Of course, Flat Stanley also works with students in biology and history, with authors chronicling the lives of collectors, with artists exploring botanical themes, and with great folks that just like to spend their spare time learning about nature. So 1 million plant specimens, eh? Does that mean that Canada has 1 million plant species? Not necessarily. In fact, we have doubles, triples, in some cases hundreds of examples of a given species. Why is this awesome? Well, if you map the location where one plant specimen was collected, you get 1 dot on a map. If you map all the locations where your specimens were collected, you get a map of that species distribution. And all animal life, including human life, depends on plants. So knowing where and how abundant those plant resources are is critical. What's more, plant distributions and abundances are changing all the time, which changes how humans do business. The fact that each specimen has not only a location, but also a date associated with it, means that people can use them to look back on past changes in distributions and flowering times, and use them to make projections for the future. And speaking of which, who knows which specimens are going to be important in the future? The plants this person saw often in the 1970s are now nationally rare. Her common plants are now critical conservation resources well after she's retired from collecting. And back when our collection began, things like DNA extraction were unthinkable, but now people take leaves from plants, or even the eggs that plant-eating insects left on the leaves, and use them to make important genetic discoveries. Yeah, technology is going to make a lot of things easier in the herbarium, but we're going to be hanging on to our flat plants. Flat plants are proof. Things like databases are someone's word for it, and we don't pay scientists to take somebody's word for it. Keeping the specimens, of course, always gives us the chance to spend time with objects that were a part of our history. We have specimens that were collected on Franklin's Arctic expeditions, so you should come by sometime and I'll show you. And at the same time, through a microscope, I'll show you lichens and mosses and microscopic algae, things that we don't notice very often in our everyday lives, but that actually make our lives possible by supporting the plants and animals that we can see. Changes in the number or the distributions of those tiny organisms in our environment can create humongous challenges for us.
So luckily, the Museum of Nature is a leader in collecting these three groups. So how many specimens is enough? I think the real question is, how well would we like to equip our successors with proof of our adventures and discoveries, giving them tools to understand their history and to manage their environmental challenges? You tell me.
Actually, I'm pretty fast. I can go. I can go with this, yeah. Anyways, what I want you to do right now is take 3 breaths. 3 breaths. One of those breaths was made by algae. One of the oxygen consumption in that breath that you took was made by algae. That is why algae is important, and that's one of the reasons why you should be very, very interested in understanding algae and microscopic organisms. This talk is set up that I'm going to justify why we should be looking at microorganisms, who are the players, what our products are, and finally what our future is. We've already done our 3 breaths. We understand that oxygen is extremely important for us and that algae are the major producers of oxygen in the world. And the second reason why you should be interested in microscopic organisms is because when you look at the basically trophic structure in the world and you try and maintain one polar bear, it takes the equivalent biomass of microorganisms of 35 elephants. I cannot put the number of zeros on top of that of how many individual algae species there would be to make that. The third reason why you should be interested in algae is pollution, water pollution. When you have a situation where the water gets worse, algae kick in and make it so that there's toxic chemicals come into the water and so on. So algae are a problem. In contrast, algae are actually good because they're great indicators of water quality. So we constantly use algae to actually A, clean the water, and B, to actually evaluate the quality of the cleanliness of the water itself. So algae overall and microscopic organisms are important. The players, Michel Poulin, he's been with the museum for 25 years. His primary interests are in marine ecosystems and biodiversity and biogeography. Myself, I'm at the museum for 26 years. I'm more interested in freshwater environments, and I'm very interested in looking at water quality, biodiversity, and general species development. The most important thing are our colleagues and our associates and our volunteers. We have currently working with over 25 research scientists around the world. We have students. We've had, over the 15 years that we've been working, we've had 15 to 20 students, and we've had a number of volunteers. And the one who just passed by, Melba Angel, is our star volunteer. We do everything in terms of tools. We use everything from drill rigs all the way down to microscopes. So even though we're microscopic, we need to use all the big tools to get the job done. In terms of what we do for overall, we look at biodiversity, biogeography, and general climate change. So we've done a lot of work in climate change, biodiversity, and biogeography. We're also interested in looking at metals and contaminants. We look at mercury right now in the Arctic environment and how it relates to algae. This is a case of cadmium, and the cadmium in terms of how it's destroying or altering cell structure. So we're very interested in metals and how they affect on the algae. We're also interested in toxins and water quality. So one of the things we're really interested in is how these metals are affecting water quality and the kinds of things that toxins that are getting into the environment. So we're also interested in blue-green algae, which is that. We've also done some work in the case of Toronto. This is a lake in Toronto, and we're looking at the history. And if you look at the history, you can say that the Iroquois, the Hurons, and then the settlers had impacts on the lakes in Toronto. Of those, it was the settlers. It was Mrs. Chambers in 1794 who had the biggest impact on the lakes, and the lakes have never recovered. We also work on ancient lakes. We work on 5 of the 12 ancient lakes in the world, and we're constantly looking for new species and evolution. The other thing that we're good at is describing new species. So over the years, we've described slightly under 100 new species and 2 new genera, and we're continuing to do that through the Arctic and also in the ancient lakes. One of the projects that we've done that everybody here has been involved in is the Rideau River Biodiversity Project. The project's been extremely productive and successful. We've had a lot of publications. We've had a State of the River report. The UNESCO designation of the lake or the river was set. And finally, we have a group still working on it. We've also done education programs. This is a simulated program with the Japanese. It's in 12 different languages, and we're using that to to try and teach about water quality and how algae fits into water quality. Over the last 12 years, Michelle and I have independently co-published 8 books, and we continue to do that. The books go from Pleistocene to present, marine to fresh, Canadian Arctic to India, and so on. And what I'd like to do is finally sum up by saying we love our job. We want to continue exploring all these things. We want to look at new species. We want to continue doing contaminants in the environment. We want to look at education and fixing up and improving our educational packages. And finally, we want to bring in new tools. So we're going to be exploring DNA and single-cell DNA extractions to bring to the museum in the future. Thank you very much.
Okay. I'm Kathy Conlon, and this is a story about gray whales and Canada. Ed Hendricks and I have worked on this, collaborating with University of Saskatchewan Fisheries and Oceans and Arcan and Carleton University. So gray whales are the only baleen whales that feed primarily on the benthos, and this is a pit that a gray whale made in the sea bottom. And this is a whole bunch of pits that's created. So they really changed the seafloor and what it looks like. Gray whales are only left in the Pacific. They were whaled out of the Atlantic 300 years ago. And the largest population is 18,000 on the North American coast and 400 on the Asian coast. Gray whales migrate from Baja California in the summertime all the way up to the Bering and Chukchi Seas. And along the way, so that's 15 to 20,000 kilometers that they travel twice a year. There's a lot of hazards for them. Killer whales will go after them, especially the babies. And also, once they're up there, they can get caught in ice if they stay too long. And this was a movie made about a gray whale that got caught in ice in the Arctic. Their feeding grounds are these red marks on the seafloor, and the one that's pink has declined over 6 years by half, and it's due to warming water caused by global warming. This is the kind of food that they eat on the seafloor. This is what I call a hotspot community. This is the real animal there, and it's an amphipod, and this is the amphipod living in its tube. And they can be very abundant, so that's why it's great for gray whales. So we were working in the Beaufort Sea actually to do with oil and gas development. This is what it looked like in the 70s, and now how they extract gas now or search for it. And our methods were working off the Nahidic, a Coast Guard ship, and we box cored, and this is what it looks like after we core the seabed. And then we sieve the animals out, make drawings of the way the community looked like, and then we map them and try and figure out how things changed. And we were collaborating with Steve Blasco at the Geological Survey, and he's mapping the seafloor acoustically. And these are all the beams of sound that bounce off the seafloor, and they pulls it all together into this beautiful, colorful image of the seafloor. So you can see the texturing. So this is an area that was venting methane. And then we overlay the animals that occurred there, and we're using GPS to locate exactly where to sample. And the ship can place itself right above these methane vents. It's amazing. And here's the drilling island from the 70s that's now only 5 meters under the seafloor. And the hole that was created or dug out to create that island. It's really impressive, the texturing of the seafloor. Well, we were out over at Cape Bathurst, and we were floored because here's that same hot spot in Canada. We never knew this, that occurs over in the Alaskan seas. And this was really exciting because it meant, well, maybe gray whales are coming. So this is a map. All the red dots are where the hot spot occurs. And it's really big. It's about 19,000 kilometers squared. So potentially this is gray whale feeding habitat in Canada. And this is caused by upwelling. That blue there happens when the wind is strong and it pushes the surface water away, and cold bottom water comes up to the sea surface. And this spreads out over the shelf. And this is what fertilizes. So on the bottom, the water is really rich in nutrients. And then when the wind is strong, it pushes that rich water up to the surface. That feeds the diatoms, and then the diatoms feed the hotspot. And that's why the hotspot can exist there. Well, gray whales actually do come there. And this is one that was satellite tagged, and it spent 18 days feeding right there, and this is right underneath the hot spots. So yes, gray whales are coming. They're using Canada. Well, this is a gray whale that got all the way over to Israel, and we figure it came through the Arctic because the sea ice is getting less and less. So the question is do you think the gray whales can actually return to the Atlantic? So this shows all the upwelling areas of the world, and what this story has done is shown us a new location. So this is an exciting story because it's maybe a positive thing for the less sea ice for the gray whales. So things are changing in the Arctic for sure. And this is a very large collaboration. All these wonderful people contributed hugely to this. And I'd like to thank the Canadian Museum of Nature for supporting us for the last decade in this study. Thank you.
Bonjour, good morning. My name is Claude Renaud. Today I'll speak to you about lampreys, and so I want you to be able to speak intelligently about them at the end of this talk. This is a chestnut lamprey. It's found in the Ottawa River. And what are lampreys? First of all, they're chordates. They have a notochord. They have rudimentary vertebrae, so they're vertebrates, and they're jawless, so that makes them agnathans. God, this is a slow presentation. Oh, here we go. This is a cleared and stained adult specimen of a lamprey, and you can see these rudimentary vertebrae with the arrows, and underneath them is the notochord. People often confuse lampreys with eels because of their general body shape, but eels have jaws, and you remember lampreys don't. They also confuse them with myxine or hagfishes because they have similar body shapes, they're both jawless, and they have round mouths. But hagfishes don't have vertebrae, so they're not. Circular mouths of doom. Okay, on the left-hand side is the hagfish mouth with its tongue everted, and on the right is a sea lamprey mouth, oral disc. How old are lampreys as a group? Here is a map of fossil localities for lampreys, and they vary between 125 and 360 million years ago. Dinosaurs eat your heart out. How many lamprey species? Only 40, but what a select and important group. The first two were described by a Swedish guy named Linnaeus 250 years ago, and myself and a group of colleagues in the last 3 years have described two others. Why are there more species in the northern hemisphere? 36 up there and only 4 in the southern hemisphere. It's because there's a lot more fresh water in the northern hemisphere, and you need fresh water if you're going to reproduce as a lamprey. Why are there no lampreys in the tropics? Very easy. The incipient lethal temperature for the amicetes or the larvae is 31 degrees, so they don't like it hot. Okay, here's a photo micrograph of a inside view of a amicete, of a larva, and this shows you the mouth apparatus on the left, and what they use to filter feed. They have a very important network as a sieve apparatus, a network of organs, and they live in the bottom substrate, the sediment of rivers. And they eat mostly phytoplankton. There's 3 types of adult feeding. There's the parasitic or the blood feeders. This is a muskellunge that was taken from the Ottawa River. It's got 2 lampreys attached to it, and you've seen past incidents of the lamprey attaching to it. Lamprey teeth, this is a cross-section of a single tooth, which you can see the cones that are cones are superimposed, the one on top of the other. 30 times in its lifetime will these cones occur. Tongue teeth in a blood feeder. This is the tongue, the piston. This is the type of plate that you need to rasp a hole through the host. And the buccal glands here are indicated. They secrete lamphredin, an anticoagulant that permits a free blood flowing into the lamprey. Now, a different type of lamprey feeding is that of a predator. So this poor Pacific herring didn't survive the experiment. The Risson River lamprey really did a good job on it. And that's the type of tongue that you get in predator lamprey. It's U-shaped and it's made for gouging. It has a central cusp that's made for gouging flesh out of the prey. Adult feeding continued. Non-parasitic lampreys, there's about 20 species. They don't feed at all for 6 months. They shorten because of that, and it's the only vertebrate that actually shrinks as it grows old. This is me in Australia some years ago, and with an electrofisher using this electrofisher to collect amicetes. Thank you very much.
Thank you. My name is Bob Anderson. I'm a research scientist in entomology here at the museum. And I'm going to talk today about the main function I serve at the museum, and that is discovering and describing new beetles. There's certainly no shortage of beetles in the world. There's about 400,000 species of beetles that are described, and I specialize in weevils, which is a group with about 60 to 70,000 described species. Mainly what I do is taxonomy. That's, as Mark said, it's the theory and practice of grouping organisms together, identifying what the species are and placing them in particular groups. In order to do taxonomy, we need specimens for study. Those specimens can be acquired largely in one of two ways. One, sort of trolling in museum collections, looking for specimens, or actually conducting the fieldwork yourself. Conducting the fieldwork ourselves is obviously the most exciting option to us. And for me, it involves mainly one of two particular things, one of which is going around thrashing through vegetation, finding what falls onto that white sheet I have, picking that up and preserving it. The other method that I've been using more often lately is something that looks at the cryptic fauna, the insects that are hidden. And that is actually digging down into the soil and leaf litter and sifting that leaf litter up. So you get a bag of debris that was basically on the forest floor. And then you hang that in these white sacks, and underneath there's a little Whirlpack bag with alcohol in it. And over the course of 3 days, anywhere from maybe 10 to 20 to 2,000 or 3,000 different arthropods fall out. And among those are a variety of groups of weevils, as you can see here. Not surprisingly, this is a fauna that is largely unknown. There are 2 undescribed genera in this photograph, and the other 2, while the genera are described, are known from very few species, but we have upwards of 100 more of those in the collection. If you look at a pie diagram, which would summarize, let's just say, the diversity of weevils, the little part of the pie that you're pulling out is the portion that is described. The big portion is the portion that's undescribed. So actually for us, finding undescribed things is more the norm rather than the rare occurrence. This is a group of weevils that I studied and published on recently, Theognetti weevils. These occur in the leaf litter. When I started this project, there were one species known from 2 specimens. We now have specimens and species from all of these different green localities. It occurs through Mexico and Central America. These are all localities where I've collected myself. We now have 95 species in this group. I just published a paper in which I described 94 new ones. The morphology is quite distinct. We're not splitting hairs with these things. You can see from this photo there's quite different external structure to them. Insects are interesting in a sense that in order to provide diagnostic identifications, you just simply have to dissect the male genitalia and have a look at it. It's like a lock and key system, so that you'll see that there's many different shapes and morphologies and all sorts of different structures associated with that. I now plan to take a look at this group of weevils, and in order to resolve these, I'm going to try and do some DNA work. The one in the lower right-hand corner, the pale brown one, I actually just described. It's the world's only known cave-adapted weevil, and it's fully blind. And a lot of these things that live down in the soil leaf litter are blind. I was just involved in a project called LLAMA, Leaf Litter Arthropods of Mesoamerica. Every good project needs a good acronym. The last year of this project, which actually was last year, we went off to Nicaragua. And I want to talk a little bit about this because this was an exciting and unexpected discovery. To this little mountain range sitting out in the middle of the Nicaraguan rainforest near Rio Blanco called Cerro Musun. It's a typical montane, wet cloud forest that you'd find scattered throughout Central America. And in walking this trail, I ran across specimens of a really interesting little weevil, which I was not expecting to get at all. In fact, when I first got it, I thought it was something totally different. If you look very closely at the head, and hopefully you can see it in this, it has a straight antenna, the little sensory appendage. And in most weevils, the antennae are geniculate or bent in its shape. And this one had straight, which told me it was very primitive. Again, when at a loss for what the thing is, we do dissections and we look at the female genitalia are on the left and the male genitalia are on the right. And comparing these with other things we know and coding all the various characters which diagnose the various groups that this thing is related to, we ended up finding that it was actually the basal member of a very, very primitive group of weevils. You can see it here as taxon 21. And everything below that forms a subfamily of weevils, and then this thing sort of fell out right in the middle. And we're actually now in the process where we'll describe this thing as a new species, new genus, and new tribe of weevil. And it was totally unexpected on this last trip. Thank you.
Microphone. Ça, c'est plutôt formel. Hein? Lorsqu'on parle de moules,
When we speak of mussels, most people think of a nice meal and a glass of wine. As a diver, I see something else. In Canada, we have terrific mussels, freshwater mussels, and we have those in salt water. Now, I'll be speaking of mussels whose shells create pearls. There are two types who have different ancestors. In Canada, we have a rich fauna. We have 6% of freshwater mussels. In the Ottawa River, we have this kind of mussel, the vital cycle. Fish are necessary for the reproduction of mussels. If you don't have any fish, the mussels cannot reproduce. I took these pictures here in the Ottawa River. You have Lampsil cordiform, and the female imitates fish to attract the fish that will distribute the embryo. My assistant and the volunteers have studied conservation, reproduction of freshwater mussels, and we also know that this kind of mussel, and I like to mention the pearls. The pearls are beautiful. Freshwater pearls can be used to make these jewels. Now, in Canada, a lot of the fauna of mussels exists. We have along the largest mussels in the world. Along Vancouver Island, you have huge quantities of mussels, and our collections are not complete. We're missing many specimens, and many specimens are badly identified. We have a lot of work to do for our collections. One of the areas I like to work is the western coast of Vancouver Island. It's a terrific place, and when you go below the surface, you find out we know very little about this very interesting fauna. Here you have a small mussel that you find in Victoria. Around the 1880s it was described, but now we see there's much more than we thought. The distribution of this species is not well known. Now, we saw pictures, and near Hideaway we found mussels of this kind. So here we have discovered a new species in Canada, and by sending divers to places that were never sampled before. So we're finding out that we know very little about mussels. One of my interests are the juvenile mussels, where the shell hasn't yet been affected by waves, by studying juvenile mussels, I hope that we'll be able to better identify the morphology of mussels and identify difficult species. Even PEI mussels are difficult to identify if you don't have the DNA. So I would like to look at the larval characteristics, the shells under microscopes, the hinges of these shells that have not yet been modified by waves. Here you can find a deposit of shells. It was discovered in 2008. And this huge quantity of shells, the blue mussels that you eat at the restaurant, exist by thousands. So I would like to compare new mussels to these ancient mussels. Now, we have a student who wants to reconstruct the marine environment of 9,000 years ago. And yes, we found pearls in these 9,000-year-old shells.
Please join me in thanking our speakers today who are very adaptable in using this new format and highly impressive. Thank you all.